Hello guys and welcome back to Excelsior Academy. So in today's lecture, we'll talk about the regulation of the citric acid cycle. So I hope we have just gone through what is citric acid cycle, and in the previous lecture, we have discussed about what are the enzymes involved and how these enzymes are converting from one substrate to the another substrate. Okay. Okay. So the citric acid cycle is regulated at multiple points. So before we proceed, just understand one simple thing. So we have got the TCA cycle. Okay. Now, if we have high concentration of ATP and low concentration of ATP, tell me whether the TCA cycle will be completed or not at this condition and in this condition. Yes, Smriti? Chandni, what do you think?
Ma'am, I'm not very sure about the answer, but yeah, in the first condition, it might be regulated because we have continuous GTP exchange.
No. See, it's very simple. हमेशा इतना याद रखो, ग्लाइकोलिसिस, टीसीए साइकिल, एटीसी, ये तीन चीज है to give you ATP at the end. ठीक है? आप खाना खाते हैं so that you get energy and so that you can work. Your cells can perform functions, right? Now, if the cell is already having high concentration of ATP, it is not going to complete the TCA cycle. ठीक है? सिंपल. क्यों मैं इतने एंजाइम्स को काम में लगाऊं जब मेरे पास वो चीज ऑलरेडी है? एक तरह से समझो ना, फैक्ट्री की तरह काम हो रहा है. You have got plenty of the product. Then why simply wasting in making more product? Why to make your enzymes to work more, right? So इस तरह से सोचो. So once you have got the more amount of ATP, you don't need TCA cycle to be completed. ठीक है? तो जब आपका ATP कम है, एनर्जी चाहिए, तब आपका ग्लाइकोलिसिस, TCA साइकिल, ETC ये सब होगा. इतना क्लियर है? Yes. Okay. So now we'll proceed with the regulation. So it is regulated at multiple points. However, in general, it is safe to say that it is inhibited by ATP and NADH. Now, why NADH? Again, we know NADH is energy equivalent. So it is around 2.5. So the inhibition by NADH keeps it tightly regulated by oxygen supply, as NADH is converted to NAD plus by oxidative phosphorylation. Okay. इतना clear है? See, if you have got high concentration of ATP and NADH, आपका TCA cycle inhibit होगा. Okay. Simple. Now, inhibition by NADH, it keeps it tightly regulated. क्यों? Because of the oxygen. And oxygen क्यों चाहिए? Oxidative phosphorylation के लिए. Okay. Now, the inhibition by ATP, it keeps the TCA cycle in balance with the energy supply. So, when ATP is high, TCA cycle inhibited. And the precursors to the TCA cycle, like pyruvate, acetyl-CoA amino acids, are diverted to other pathways. And what are the other pathways? जब कोई चीज आपके पास ज्यादा है, ठीक है, तो आप क्या करते हो? स्टोर करके रखते हो. So all these precursors will be directed towards your gluconeogenesis. जब मेरे पास बहुत है resource, what will I do? I'll just collect and keep it for the further use, future use. है ना? Guys, clear? Okay. Now, acetyl-CoA, citrate, succinyl-CoA, they are the end products of the individual steps in the citric acid cycle, and their accumulation inhibit the step involved in their production. Obviously, right? ये क्या होगा? इसको हम क्या बोलेंगे? Feedback inhibition. This is called feedback inhibition. So what does it mean? It means once you have got the product in large concentration or high concentration, they are going to inhibit their own enzyme to stop the further synthesis. Okay. So that of course results in the inhibition of the cycle as a whole. Finally, calcium 2+ stimulates the citric acid cycle at several points. Now this is important because electrical stimulation of the muscle causes increase in the intracellular calcium level. Thus, During exercise, the TCA cycle will be maximally stimulated in the muscle. So what does it mean? So guys, whenever you're using your muscles like contract, relax, आप कभी कुछ काम कर रहे होते हो, so what will happen? In the muscle, the cells, they are continuously undergoing glycolysis, TCA cycle and ETC because every time the energy is required to contract, to relax, to contract and relax. है ना? आप जब बात भी कर रहे हो, हाथ हिला रहे हो, तब भी आपको जरूरत है. कुछ काम कर रहे हो, while you're driving, every time you can see that your muscles are always in action, unless until the body is sleeping, right? So every time you are going to have the calcium levels high, and it is going to activate your citric acid cycle continuously. Is that clear, guys?
Yes, ma'am.
Simple. Now regulation of the citric acid cycle. तो अभी यहाँ से आपको पता चल जाएगा. So plus means activate कर रहा है, minus means उसको inhibit कर रहा है. ठीक है? अगर ये cycle याद हो जाएगा, पूरा आपका regulation आपको याद हो जाएगा. It's very simple but complicated, of course. So you have got the pyruvate. Okay. Now you have got the acetyl-CoA. Now acetyl-CoA, if it is in large quantity, what will happen? It is going to inhibit its own enzyme. राइट. यहाँ पे एक एग्जांपल हो गई आपका फीड बाय इनहिबिशन का, जो हमने अभी जस्ट डिस्कस किया. ओके. सो नाउ oxaloacetate acetyl-CoA with the help of the citrate synthase, it is going to give you citrate, right? See, अगर आप यहाँ पे नोटिस करो, हर एक सबस्ट्रेट अपने प्रीवियस जो अपना एंजाइम है उसको वो इनहिबिट कर रहा है. इफ यू सी केयरफुली, है ना? So more concentration of citrate is going to inhibit its own enzyme. Is that clear, guys? Then ATP is also going to inhibit the citrate synthase. We need to understand in such a way that कल को जब भी कोई क्वेश्चन आए इस पे बेस्ड, so you should be able to answer. Like suppose options में दिया है आपको ATP दिया है, citrate दिया है, and when you talking about citrate synthase, so both of these are going to inhibit your citrate synthase if these are in the high concentration. Clear? Okay, of course. I mean NADH भी है आपका. So if you see dotted lines जो है वो सब आपका inhibit कर रहे हैं, and जो dark line है वो आपका activate करने वाली चीज. Clear? इतना clear हो गया first enzyme के बारे में, citrate synthase. So you have got the ATP, citrate as well as NADH to inhibit the citrate synthase. Next, citrate to isocitrate, isocitrate to alpha ketoglutarate. Now alpha ketoglutarate to your succinyl-CoA. So guys, you can see over here, जब भी हमारा alpha ketoglutarate succinyl-CoA, we know that over here is dehydrogenase enzyme, right? So here you are going to have the NADH production. Okay, so NADH क्या करेगा? यहाँ पे भी आपको inhibit करेगा, because I don't want more concentration of alpha ketoglutarate. Okay, और कैल्शियम क्या कर रहा है? इसको एक्टिवेट कर रहा है. So everywhere you can see that जहां पे भी आपका TCA साइकिल इनहिबिट हो सकता है, वहां पे कैल्शियम आपका एक्टिवेट कर रहा है, एंड सबस्ट्रेट अगर हाई कॉन्सेंट्रेशन में है तो उसको इनहिबिट कर रहा है. इतना क्लियर है गाइस? यू कैन सी दैट ATP इज आल्सो इनहिबिटिंग. ठीक है ना? succinyl-CoA is going to inhibit because succinyl-CoA to succinate we are going to get GTP. तो यहाँ पे अगर इसका कॉन्सेंट्रेशन ज्यादा हो जाएगा तो ये भी इसको इनहिबिट करेगा. करेक्ट? एंड देन यू हैव गॉट दी, ये आपका सिंपल जाएगा. तो आपका रेगुलेशन कौन कौन से पॉइंट में हो रहा है? फर्स्ट स्टेप में हो रहा है जहाँ पे oxaloacetate acetyl-CoA है, regulating citrate synthase. Then over here we have got alpha ketoglutarate, यहाँ पे dehydrogenase succinyl dehydrogenase के पास भी आपका कर रहा है. ठीक है? इतना क्लियर है गाइस? दिस इज वेरी 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 इंपॉर्टेंट स्लाइड. अभी यही चीज हम थोड़ा सा डिस्कस करते हैं, लाइक व्हाटएवर थ्योरी इज गिवन. So citrate synthase reaction. Citrate synthase is controlled by the concentration of acetyl-CoA, which in turn governed by the activity of pyruvate dehydrogenase complex. Now, Smriti, try to tell me why we are saying that it is governed by the activity of pyruvate dehydrogenase complex when we are talking about citrate synthase. क्या हो सकता है connection? Chandni, can you give the answer? Yes, anyone?
Ma'am, because it was controlled by the concentration of acetyl-CoA.
Yes, yes, very good. Because pyruvate dehydrogenase does what? It converts your pyruvate to acetyl-CoA, right? Good, good again. So now, ATP is an allosteric inhibitor of citrate synthase. What does it mean? So citrate synthase is also having another site at which ATP can bind and inhibit the citrate synthase activity. Okay. So ATP increases the Km for acetyl-CoA, thus reduces the affinity for the enzyme towards the acetyl-CoA. So as we remember from our previous lectures, high Km means low affinity. Okay. Now how it is increasing the affinity? By binding to it and making it having less affinity for the citrate. Sorry, the acetyl-CoA. Now, high acetyl-CoA levels also decrease the affinity of citrate synthase towards the acetyl-CoA. Citrate is a competitive inhibitor of oxaloacetate on the enzyme. Now, guys, can you tell me over here? Citrate is there, oxaloacetate is there on the enzyme. So, how can you say about this? You know what is competitive inhibitor? Citrate is actually a 6 carbon and oxaloacetate is 4 carbon. Now, how it is going to compete for the enzyme? Yes. कैसे होगा? So you know what is competitive inhibition, right? It is targeting the same active site. Yes, Priti, Chandni, any explanation for this?
Citrate increases its concentration, ma'am,
So what will happen when there is an increase in the concentration of citrate?
and competes with oxalic acid.
Yes, yes. So see, it's very simple. In a competitive inhibitor, if you have got more citrate, so what will happen? Since there is an active site, and if you see the structure of the oxaloacetate and citrate, you will see that it is able to bind to the active site of the enzyme. So it is going to act as an inhibitor. So what will happen? If the concentration of the citrate is getting increased, obviously the concentration of oxaloacetate is decreasing, right? And that's how it is able to inhibit the enzyme. So you can stop the TCA cycle at the very beginning. In the first step itself, you can stop the cycle. Okay. So isocitrate dehydrogenase reaction. This reaction appears to be the rate limiting step of the citric acid cycle. So it is allosterically stimulated by ADP, whereas NADPH is an allosterically inhibitor. Again, same thing. अगर आपके पास ADP ज़्यादा है, it is going to activate or stimulate the cycle. If you have got the NADPH more, it is going to inhibit. Simple. Now, alpha ketoglutarate dehydrogenase reaction. Succinyl-CoA, NADH, they inhibit the activity of the alpha ketoglutarate dehydrogenase. Why? Because this is responsible for the conversion of alpha keto to succinyl-CoA, right? So now here the succinyl-CoA is acting as a competitive inhibitor for the coenzyme A, which is a substrate for the alpha ketoglutarate dehydrogenase. ADH means alpha ketoglutarate dehydrogenase. So again, double barrel effect over here. So again, the accumulation of the succinyl-CoA will increase. It will act as the inhibitor, and simultaneously, what will happen? The concentration of the CoA will get reduced. So again, it is able to inhibit the TCA cycle over here. अगर आपसे first step में नहीं inhibit हुआ, so in the second step it is going to inhibit the cycle. Okay. Now this is really very important. Like you need to understand about the anaphylortic reactions. What does it mean? Anaphylortic means it is filling up the reaction. There will be gap create हो जाते. For example, sometimes you don't have the substrate, okay, but you need to continue the cycle. So that cycle gap filling is done by these reactions. Okay. So कौन करता है? So TCA cycle act as the gap filling reactions for the other cell cycle to continue. Okay. ये एग्जाम में आ चुका है. So this you need to remember. So many intermediates of the TCA cycle are used for the synthesis of the other substance. So they act as the gap filling substrate reactions for the other cycle. Okay. So that those other cycle can continue. So anaplerosis is defined as any reaction that can restore the concentration of a crucial but depleted intermediate. See, this is why I'm saying TCA cycle is very important. क्योंकि वो क्या कर रहा है, दूसरे cycle को continue करने के लिए it is giving its own substrate over there. Okay? Several biosynthetic pathways utilize TCA cycle as the starting material, and that's why it is also called amphibolic, यानी कि both anabolic and catabolic. Now, justify your answer why TCA cycle is known as the amphibolic cycle. This is your hint. Why it is both anabolic and catabolic?
Because it is responsible for the synthesis of new reactions also in the cell, as well as it is going to yield the end products that can act as synthesis starting products for another corresponding reaction.
That is
or
anabolic.
cellular pathway.
Like it is giving the starting material to the other cycle. तो वहाँ पे वो anabolic हो गया. Catabolic मतलब क्या? कोई भी कंपाउंड का डिग्रेड कर रहे हो, ठीक है? तो यहाँ पे क्या हो रहा है? ग्लूकोज से आपको एसिटाइल कोई मिला, उसको आप और छोटा करके एट द एंड एटीपी जनरेट कर रहे हो. So you're metabolizing over there. You're using that particular compound. So that's why it is both anabolic and catabolic. अभी क्लियर है ना? नहीं? Yes, Prati?
Yes, ma'am.
Yes, ma'am.
Okay. So the TC cycle in the liver is often known as open cycle because there is such a high influx of the intermediates. After a high carbohydrate meal, citric efflux and cleavage facility provides acetyl units for the cytosolic fatty acid during the fatty acid synthesis. So during fasting, this gluconeogenic precursors are converted to malate, which leaves the mitochondria for the cytosolic gluconeogenesis. So this is what happens, guys. जब भी आप कभी ज़्यादा खाना खा लेते हो ना, तब आपको नींद आती है. ओके, नाउ नींद क्यों आती है, कोई मुझे बता सकता है? Why do you feel sleepy after having a heavy meal, especially if you eat rice? Why do you feel sleepy?
Ma'am, because the stored oxygen, which will be actually used for like ATP synthesis somewhere else, will get used up during the carbohydrate metabolism.
Almost correct. You need to add more in this. See, what you have to say is that when you eat more, of course you need to digest more, right? And you need more. Digestion means क्या? आपकी cells काम करना है, right? It needs to take those digestion glucose also for the storage or for the tissues or for the cells. So even for that, and that storage is possible only when the glucose is being transported with the help of the blood to the different parts of the body, right? So what happens is that the blood circulation or the brain does not get that much of glucose क्योंकि सारे आपके blood pumping जो है towards the digestion में चला जाता है. Okay, so then they start focusing on the digestion, and that's why your brain gets less amount of energy and you start feeling sleepy. अगर आप नहीं सोओगे, what will happen? You will feel heavy, right? You are not able to work as efficiently as you can do when you are not feeling sleepy, है ना? इसलिए कुछ लोग क्या करते हैं? They take this caffeine का shot. Either they will have tea after eating lunch or they will have coffee. So especially it happens when you are at the job. Of course, you can't sleep over there after having lunch. So then immediately they go for the caffeine. So what caffeine will do? It is going to boost up your nerve impulses. Okay, like serotonin, yeah, अच्छी चीजें जो होगी, like good neurotransmitters. That's why it keeps you active even after having a heavy lunch. Okay. So what will happen then? So whenever now you eat heavy lunch or heavy dinner, anything, it has to be related to the carbohydrate only. So whenever you have such things, you can just imagine that if you're sitting idly, sleeping, the body is converting the entire carbohydrate into fat. And that's how people become fat. जो कुछ काम नहीं करते हैं, खाना खा के सो जाते हैं, उनका like first of all weight will come out, stomach will come out, right? So that's how people start becoming obese. because they eat and sleep. And many times you must have heard from your mother also or your grandparents saying that खाना खा के मत सोया करो immediately. This is the reason, because it is going to convert everything into your fat. Clear, guys? So it's not necessary that you eat oil to become fat. If you have carbohydrate only in your meal, that is sufficient to make you fat. Okay. Okay. So we'll go slowly because there's so much to understand from this. So some important anaerobic reactions of the TCA cycle are intermediate are used for the synthesis of the following substrate. So सबसे पहले क्या हो गया आपका pyruvate carboxylase. So where we use pyruvate carboxylase? कहाँ पे आता है? Who can answer? Chandni, who can answer me? Pyruvate carboxylase. It is in the gluconeogenesis. Okay. तो जहाँ पे आपका pyruvate kinase होता है to convert phosphoenol pyruvate to pyruvate in glycolysis. Okay. Now, if you go to the gluconeogenesis, वहाँ पे आपका instead of pyruvate kinase, you have got the pyruvate carboxylase. Okay, so the citric acid cycle operates when the oxaloacetate is available. In animal tissues, especially liver and kidney, oxaloacetate is formed by the carboxylation of pyruvate by pyruvate carboxylase in mitochondria, and it is reversible, but this enzyme is absent in mammals. So what is happening? Oxaloacetate जो है, it is formed by the pyruvate carboxylase enzyme. तो आपने क्या किया यहाँ पे? Oxaloacetate is formed carboxylation of pyruvate. क्या किया आपने यहाँ पे? Carbon molecule add किया है. Okay. Now, pyruvate carboxylase is homotetramer. Each subunit of carboxylase contains active site that are tightly bound to the Mn2 plus and Mg2 plus cofactors and covalently bound with biotin, and also an allosteric site which binds to the acetyl CoA. Wow. So you have got the pyruvate carboxylase. It has got the Mn2 plus या Mg2 plus का binding site. Okay. It has got a site where the biotin is bound, and it has got a site where your acetyl CoA can come in bind. Okay. And biotin is attached to the pyruvate carboxylase with the help of the lysine. It also requires a monovalent cation K plus apart from the divalent cation. Okay, because we had K plus involved in. Now, what does pyruvate carboxylase does? It adds CO2 to the pyruvate to form oxaloacetate. This is important. Now, the biotin of the pyruvate carboxylase enzyme, it forms a covalent intermediate with CO2 in reaction requiring ATP and Mg2 plus. So, guys, यहाँ पे important है क्यों? Since it's a gluconeogenesis, right? It's a gluconeogenesis. So, you are going to use ATP. ATP usage होगा यहाँ पे. So that's why यहाँ पे हमने क्या बोला? Biotin क्या करेगा? CO2 के साथ covalent bond बनाएगा, which will require ATP. ठीक है? Now, this activated CO2 is then transferred to pyruvate to form the carboxyl group of the oxaloacetate. Now, what is later what is going to happen? Okay. So pyruvate carboxylase is activated by acetyl CoA, inhibited by the high concentration of acetyl CoA derivatives. So, if suppose you don't have oxaloacetate, the rate of citrus in this reaction decreases and the acetyl CoA concentration rises. क्यों? See, आपके पास oxaloacetate plus acetyl CoA gives you citric. अभी आपके पास oxaloacetate नहीं है, तो क्या होगा? Citrate नहीं बनेगा. This will result in the increased concentration of acetyl CoA. Correct है? है ना? And then it is going to activate pyruvate carboxylase to synthesize more oxaloacetate, so that your TCA cycle can be continued. Guys, समझ में आया कैसे हम इसको रेगुलेट कर रहे हैं?
Yes, ma'am.
हाँ, याद रहेगा. See, बहुत सारी चीजें हैं इसमें. What you need to remember is, let us summarize the slide now. I'll just summarize the slide for you. So you have got the pyruvate carboxylase. ठीक है, ये आपका gluconeogenesis का enzyme है, the first enzyme. It has got K plus binding site. It has got divalent Mn2 plus and Mg2 plus binding site. It has got biotin. Okay, and it has got a site where your acetyl-CoA can bind because it needs to activate or inhibit the enzyme. ये हो गया आपका पायरुवेट कार्बोक्सिलेज के बारे में. इसका काम क्या है? To convert pyruvate to oxaloacetate. Convert your pyruvate to oxaloacetate. Okay, now simple. कैसे करता है पायरुवेट कार्बोक्सिलेज? इसका बायोटिन which is attached to the pyruvate carboxylase with the help of the lysine makes a covalent bond with the CO with the help of the ATP. Then it is transferred. This COO group is transferred to the oxaloacetate to form the oxaloacetate. ये हो गया इसका function. Okay. Now what happens is that if you have, see, we know that in the TCA cycle, oxaloacetate and acetyl-CoA gives you citrate. Okay. If you don't have oxaloacetate, citrate synthesis will be stopped or inhibited. This will result in the increase or concentration of the acetyl. Now, जब ये आपका ज़्यादा हो जाएगा, it is going to activate pyruvate carboxylase to synthesize more of oxaloacetate. अब इससे क्या होगा? Finally, now you have got the oxaloacetate, acetyl-CoA. Now the TCA cycle is active. अब आप citrate बना सकते हो. ठीक है? Same thing is over here. जो भी हमने डिस्कस किया, सेम चीज यहाँ पे है. It's just that we have also added the, yeah, we are going to talk about the amino acids. So बिना amino acids के, if you see this, it's same thing which we have just discussed now. हम इसको और करेंगे more when we talk about the amino acids. Okay guys, amino acids के बारे में हम next lecture में discuss करेंगे. आज हमने जो main topic था हमारा pyruvate carboxylase का था, like how it is helping in the regulation of the TCA cycle. Okay, आज का क्लास क्लियर है गाइस?
Yes, ma'am.
Yes, ma'am.
ठीक है, तो क्वेश्चन आंसर्स कर लेते हैं. लेट्स डू सर्टेन न्यूमेरिकल्स एंड नॉट न्यूमेरिकल्स, क्वेश्चन एमसीक्यूज. वेरी सिंपल एमसीक्यूज लाइक जो मुझे प्रीवियस क्वेश्चन पेपर से मिला है. सिंपल. Krebs cycle takes place where? Okay, the meeting time is getting over. Just join me back again, guys.